that's in at BIL. There's a couple members of the BIL team that are on the line right now that will be speaking in a little while. One is Mariah Carey, and she'll introduce herself when it's time to speak. And the other is Luke Tootie, and he will be speaking momentarily as well. For those of you who are interested in the agenda today, we're going to start off. I'm going to give a brief introduction to the brain image library, including the data scope that we have. Then I'm going to turn it over to Luke, who's going to talk about the using the BIL portal, some information about file organization, and using on demand as well to look at your files. We will have a break and then turn things over to Mariah, who will talk a lot about metadata and submitting data that you have to BIL and the metadata models that are in use. So with that, let's get started. So about the brain image library, we are a public resource serving the neuroscience community by providing a persistent centralized repository for brain microscopy data. The brain image library relies on contributions from brain researchers like yourself, and much of our current data comes from investigators that are part of the BICCN and BICAN networks. NIH funding is not required to contribute data to the brain image library, but the data must be of interest to the Brain Initiative. And since many of the data sets that we house at BIL are quite large, we provide an extensive analysis ecosystem where users can explore the data in place without downloading it at no cost. We also provide complementary network path analysis to sites experiencing network issues, either uploading or downloading data to us. So what's our data scope? So our data scope includes whole and partial brain data sets from mice, primates, including humans, and model organisms. Targeted microscopic enabled experiments, including things like connectivity between cells and spatial transcriptomics, as well as historical collections of value to the community. To give you a preview of some of the high quality data in BIL, this image shows a cell population data set contributed by Dr. Pablo Austin, formerly at Cold Spring Harbor Laboratory, now at Certigo Therapeutics. This 2-channel whole brain data set, which is about a few hundred slices, is actually one of the smaller data sets that we house at BIL. Some of the largest, highest resolution mouse data sets that we currently have at BIL are generated using the FMOS technology. These FMOS data sets range from about 8 to 13 terabytes each and contain about 10 to 15,000 slices per data set. The resolution of each individual slice is about 35,000 pixels by about 55,000 pixels with a Z spacing of about 1 micron. I also want to note that some of the largest data sets that we've been receiving recently are actually spatial transcriptomics data sets from MERFISH. And I believe that our largest data set that we house is somewhere around 100 to 150 terabytes. In terms of existing coverage in BIL, the overwhelming amount of data sets contributed to BIL are from mice, with relatively fewer data sets from macaque, marmosets, and humans. Though we expect as the BICAN effort gets ramped up, the amount of data sets that we will receive from primates and particular humans will increase. In terms of data modalities, most of our primary data sets are slice-wise image stacks from whole brain imaging experiments. In addition, we also have thousands of tracings from primary image data sets that have been deposited in the archive. In terms of technologies, we have data from most microscope types, as well as microscopy data that has been produced by multimodal experiments. In terms of data, we've been roughly doubling in the number of data sets that we receive about every year. Currently, we have over 15 million public files that are available. So next, I want to briefly mention some of the additional services that we provide to data contributors. These services include network path analysis and receiving data via alternate media in cases where the last or the networking path really has insufficient bandwidth. We also issue DOIs for our data sets and can issue DOIs for collections of data upon request. Next, I want to briefly mention the resources that we provide to explore data through our analysis ecosystem. This includes both small and large memory nodes, nodes with GPUs, as well as our Bridges 2 and Cerebrus CS2 high-performance computing systems. BIL also provides a remote desktop environment and has the capability to host unique web interfaces to the data, as well as supporting numerous software packages. So one of our big efforts going forward this year and into the future is an effort to make the data that is deposited in BIL more accessible. Part of this effort, we are developing an on-demand transformer that will serve the data that is in BIL in a variety of formats, including visualization formats that will plug in directly to NeuroglanCer and Napari as well. We also plan to make accessing BIL easier by using the access mechanism to allocate our resources. And that will allow for easier administration to allow folks contributing data and using our data to work together in an easier fashion. And we also will be working a bit more on our metadata for usability and greater accessibility, including developing links to outside resources. So I'd now like to briefly go over how you can get started depositing data in BIL. So ideally, before you submit your proposal, contact us at bilsupport@psc.edu to just discuss your project a few weeks prior to submission. Including things like, we would like to discuss the anticipated types and amounts of data that you're going to be producing, as well as what your anticipated submission timeline would be. If you contact us, we can provide you with a letter of support stating that we will receive your data, as well as provide you a template NIH data management and sharing plan that outlines the specific standards that are in use at the BIL archive. And we are also always open to collaboration and would be delighted to discuss any potential collaborations with you beyond simply just receiving the data that you're providing, and would be glad to discuss anything with you at that time. So once you've received funding for your project, first of all, we would like to congratulate you because not every proposal is funded. So you've made the pass, and we're excited to receive your data. We would like you to contact us again in greater detail to discuss the amount of data that you're going to produce and what your anticipated submission timeline is. A lot of times when proposals go in, if they receive a fundable score, sometimes there are budgetary adjustments which also adjust the amount of data that can be produced. So it's important that you follow up with us so that we know that you're a funded project, as well as we know how much data you're going to be giving us and when you're going to approximately be giving us that data, just so that we can plan our systems and make sure that we have the space needed to accommodate your data. With this discussion, we will also advise you on the current metadata template that is most appropriate for the type of data that you're going to be contributing. Importantly, how to structure your data to ensure that it can be visualized through our on-demand transformer system. And we also can give you more exact instructions on how to gain access to the BIL systems, including our analysis ecosystem, as well as any online upcoming training that we are offering. So that's all great. When you're ready to submit your data to us, we encourage you to plan ahead and start your data submission process well in advance of any consortium or publication deadlines that you may have for making the data public. Please consider the time required to move data as well. As data sets get larger and larger, it does take a considerable amount of time to move the data. If you do run into problems transferring the data, again, I want to reiterate that we can provide assistance locating where the transfer bottleneck is occurring. Sometimes we can resolve these issues, such as if the data is being routed incorrectly. Otherwise, sometimes we can just pinpoint the location on campus where you'll have to work through the issues with your local campus IT folks. When we receive your data and you send us a request to make your data public, we validate that data. And I do want to point out that the data validation times does vary quite substantially, and it will roughly be in proportional to the number of files in the submission. So if you can structure your data such that you can have a smaller number of larger files, that would be preferable over a large number of small files. And in general, smaller submissions are preferred because we can validate them faster. So if you have 10 data sets, you can submit those all in one submission, or you can submit those as 10 submissions. So if there's data that you need to get through right away, smaller submissions that you can let us know to process while you're uploading your other submissions would be preferred. And we also did want to mention that we do curate the data, particularly the metadata that we receive, and there have been some occasions where we need your assistance to resolve issues with your metadata. If such is the case, the curation team will get in touch with you, and we do need your assistance in those situations to resolve those issues. So that's all I'm going to talk about to introduce you to BIL. I'd be happy to answer any questions now or at the end of the day. You should also feel free, I should mention that if you have specific questions, you can also feel free to ask those questions in the chat. And as we see them, we will address those issues in the chat as well. So with that, I'm going to stop sharing my screen and allow Luke to share his screen, and I will turn over the next portion of the workshop to Luke.
Just want to make sure if I'm sharing the correct screen. Can everybody see a slideshow?
Yes.
Okay, awesome. As Alex mentioned, hi everyone, my name's Luke. I'm a web developer with the PSC, primarily on the Brain Image Library project. I work on the data submission portal as well as data processing for the incoming data that people submit. Today, I want to talk about actually how you would go about submitting data and all the steps you need to do to get there. So yeah, today I'm going to go over the workflow for submitting to BIL, how to create a submission, structure your data, doing a file transfer up to BIL. I'll hand it over in a little bit to Mariah to talk a bit about metadata, and it'll come back to me and I'll show you how to submit that metadata. And then I'll touch briefly at the end here on some of the functionality we have for PIs through the portal and the exclusive dashboard that PIs and data managers are provided through us. So yeah, the first thing I'm going to talk about is the first thing you'll need to do to actually get onto BIL is to request an account. I believe everybody here has an account. If not, that's okay too. Please feel follow along the best you can. But if you are going to create an account at BIL, the first thing you need to do is visit the Access Portal website. This website is where you'll fill out some information and get back with a login for the Access portal. Those links are located here and here, as well as on our brainimagelibrary.org website. So this is just showing after you do that, you'll be sent back an email basically saying that your Access account has been approved. Go ahead and create your password at PSC and send a ticket to the BIL support line at billsupport.psc, or sorry, billsupport.psc.edu, or yeah, at BIL support, and send a ticket there and let us know that you have an account and you want to submit to BIL or get access for either submitting or just kind of exploring data too. You can do that as well. And it's just showing, a few other things you'll see when you create an account. So once you've gone ahead and created an account and you have access to BIL and you're able to log in, if you're submitting data, obviously the first thing you'll want to do is to create a new submission. So the submission portal is located at submit.brainimagelibrary.org. It's the portal that allows you to create a submission. It grants a landing space on our hardware where you can point an upload to and deposit your data to. And it gives a detailed view of the data that you submit. So I'll show a hands-on here on how one would go ahead and do that. On the BIL submission portal, there is a drop-down menu for create a new submission and then a form. I'll go over on my other screen here and share actually going and doing that hands-on. So if folks go over and type in submit.brainimagelibrary.org, you'll likely be prompted with a login. If this is your first time logging in, that's fine too. Your dashboard will look a little bit different than mine. I have access to the PI dashboard that I'll show later, but it still operates exactly the same. You can still create submissions, add metadata, view your submissions, etc. So if I want to go ahead and create a submission, I'll go to new submission. So here's the form to create a submission. Obviously, the more descriptive you can be, the better. For the sake of the workshop, I'm just going to be a little generic. So I'm just going to say this is the summer workshop submission. My description, I'll say test workshop submission. I'm with the PSC. My lab name doesn't really matter for this. I'm just going to say the PSC as well. Project funder ID, your project funder ID. So the grant number that you're associated with for submitting data. For me, it doesn't really matter. Again, we're just doing a test case here. Down here on project, if you click this, everybody should have an option that says their username project one. This serves as a scratch space for testing out the portal and seeing how things work. If you haven't yet been set up with the project you're going to be submitting data for, so specifically the grant project that you want to submit for, let us know either via BIL support or the chat, and we can get that set up at some point. But for now, just because this is a test, we'll just use our scratch base project. So if you click that, there should be an option for that. If you click that, it'll drop in here a number. This is just the internal number that we use. Don't worry about it. Just click it. And if you hit save, it's going to prompt you over to upload a metadata spreadsheet. Now we're not there yet. So if you go back up to view and then submissions, I have a lot here, but you folks should just see one, and that's the one we just created. Mine is called Summer Workshop Submission. I'm going to click on that just to get a better detailed view of this. And I'm going to flip back over now actually to my slideshow to explain a little more. Oops. If I can find my screen. So this is just going over what I just said. So creating that submission and getting this landing page on your browser. If anybody's having trouble, let us know. Has everybody been able to do that so far? If everything's good, I'll continue to move on. So as I was saying, you have a new data submission and it's been created. If you take a note on that screen, you'll see a field there called data staging area or data path. This is where you're going to point that upload to. So, and I'll get to that here in a moment. But this is the staging area at BIL where you can upload your data to in kind of a pre-public area before official submission. So I'm going to show here actually going ahead and uploading a piece of data to that directory we just created. There's a lot of options for uploading data to BIL, and it really comes down to A, what are you most comfortable doing, and B, what works best for your team. There's a lot of different protocols and programs you can use to point a data transfer to somewhere. If you're using Windows, you can use SFTP or SCP. You can do that in command line, or if you're more comfortable with using a user interface, there's a few programs such as WinSCP and FileZilla. There's plenty others too. Those are just the most popular. You can use those for those kind of file transfers. There's also Globus, which is a popular one because it has a really nice user interface and is it's just easy to use. I'll do an example of that as well. Same situation for Mac pretty much. They can use FileZilla and Fetch, and they can also use rsync, which is a strictly command line way to transfer files. Globus is also available on Mac. And if you're using Linux, you're probably going to be stuck using more of command line options. So here's an example of somebody doing an upload using rsync. As you can see, this is in somebody's terminal. They're running an rsync, which is a built-in command to Mac and Linux. A few flags set that we recommend. Mouse one in this circumstance would be a folder that they would like to upload to their landing zone. And then you can see here, test user would be your username at upload.brainimageslibrary.org : and then here, BIL landing zone, your username, and then that data staging area UUID that we made note of earlier. So if you're interested in using Globus for file transfer, here's a step-by-step on how to do so. You'll go to the cilogon.org. I'll click this and show what information you'll want to gather here. It's going to ask you to log in or actually log out to show you. If you go to the selected identity provider, and if you scroll down to or search access CI, which will say exceed in parentheses, you can log on using that, and this will authenticate with your access information. Should prompt you for a 2-factor on your phone, and it'll have you set that up when you're doing the access account creation. So if you're interested in using Globus, the information you'll need to send to the support line will be this certificate subject, as well as your EPPN here. So it'll pretty much just be your username at access.ci.org and this certificate subject. Just send a ticket and say, I'm interested in setting up Globus for a connection to BIL. We'll know what to do from there. So yeah, just provide all that information to us and send it over to BIL support. And we'll send you back information on how to get set up fully with Globus if you want to use that. This is what the Globus interface will look like. Under File Manager, you can search for our endpoints, and that would be Brain Image Library. There's two options. There's one for downloading data, and there's one for uploading. The second option here, Brain Image Library Upload with CI Logon, is going to be for uploading. You can feel free to use either to upload or download, whatever you please. Looks a little bit something like this. On the left side of your screen will be your local machine, and on the right will be the BIL file system. And you can see here, you can point that data path in this path right here and select the files or folders you'd like to upload and hit start. It's pretty nice because it'll give you a timeline of how long it's going to take to upload, and you can close out of your screen and it'll still continue doing it for you. And here's another example I wanted to show for people who are interested in using SCP or SFTP. This is very similar to an rsync command. In this example, they're doing SCP, which is the file transfer protocol, a file, and then they're just uploading it to their directory. And now this is another example of a user interface for WinSCP. This is that for SCP. This is that WinSCP program I was talking about, kind of similar to how Globus looks. It's easy to use. So whichever you're most comfortable using, feel free to use. We're open to support for most anything for file transfer. I'm going to show an example of actually creating a file and uploading that to our directory we created. So we're just going to create a dummy text file. You can do this any way you wish. I'm going to show an example just doing it in terminal just quickly. You can make a text file on a notepad if you want to. Anyway, doesn't matter. So I'll switch over here and just show me doing that and pointing an upload to my data directory. So here I'm just going to do cat. I have my terminal open here. I'm just going to do a cat myfile.txt. I'm going to type, this is my new file. And then Command D will close that. Oops. Control D rather. So I just have an empty text file here. I'm interested in uploading this to my data collection at BIL. So I'm going to hop back over to my portal here just to copy my data path. And again, that's right here. So in my terminal, I'm going to use an example of SCP. I'm going to do SCP myfile.txt, my username at upload.brainimagelibrary.org, and then you do colon, and then do colon, excuse me. Upload.brainimagelibrary.org, colon, and then I don't think I got that copied correctly on my end, so let me just grab that again. There's my data path. So I'm going to hit enter. Oops. Looks like I need another slash there. I think I'm following my own directions wrong here. I apologize. We may be having something technically going on here. Alex or Ivan, are you online? Is there something I'm doing incorrect with my?
It looks like you have library misspelled, Luke.
Oh, okay, that'll do it. Sorry about that. There we go. So it'll prompt you for your password. So that uploaded my file up to BIL in my landing zone directory. I'm going to jump back over to my presentation here. So again, it's showing that with our file created, we can upload it. This is showing that example. Feel free to type library correctly. I apologize for not doing so. Once you do an upload to BIL, there's a few different ways you can actually look at the files you uploaded. Some people prefer to do it primarily in the command line. That's fine if you'd like to do so, but on the BIL submission portal, we actually have links to our on-demand program, which Mariah will go into a little more depth later. But for this example, I just want to show that it's pretty easy to just take a look and see the files you uploaded and move around things, edit, restructure, however you'd like to do. I'll share my screen over here again on the portal. This is a fairly new feature we just put on here too, with the on-demand program. So I'm going to select my submission I did. And there's an option down here that says view submission directory. If you click this, this will jump over to on-demand. It'll ask you guys to authenticate if you've never done so, but it just shows that it just shows the data directory on BIL, and we can see that file got uploaded there. Again, you can use this to move things around or restructure or add directories, however you'd like to structure your data. So yeah, you can look at it that way. This link too, I should mention, when your data is not public, it'll link over to the landing zone directory. Once you've gone through the entire submission process, this will change to a public directory on the BIL file system as well. So you can easily get to your landing zone or when your data is public that way, if you like. Back over here. So yeah, this is just showing another screenshot of that. This is a directory that I had empty, but if you just click over to that, it'll show what you have on the file system. With that, at this point, you've uploaded data, you've created a submission, you're ready to go ahead with providing metadata to us. I'm going to jump that over to Mariah, who's going to talk about file organization and metadata.
Thanks, Luke. So before I get started, I just want to see if everyone is kind of up to speed. Have you all been able to at least log in and see the submission portal? Can I get a thumbs up if you're following along and you are able to access all of these tools? There's no questions. I will go ahead and share my screen and talk a little bit about file organization. So can everyone see my presentation?
Yes, Mariah.
Yes, thank you. All right. So now that you know where to upload your data, what you also need to know how it needs to be organized in order for it to be properly ingested and published at BIL. And so, you've gotten through all of these steps in the submission process. So once you're ready to upload everything, how are you going to structure it? In order to answer that question, let's talk a little bit about the definition of a data set and a submission. So we'll talk about the metadata a little later, but you will be entering in metadata specifically for every single image set, which can be, an image volume, a multi-dimensional image that is associated with a single subject or a single experimental unit. I like to think of that as, maybe you've had a specimen that you've imaged multiple times using different parameters. So anything that has a unique set of metadata associated with it is called a data set. You can have many data sets inside a single submission. Typically, data sets contain multiple image files for 2 or 3 dimensional volumes. So the submission is going to contain, it can contain one data set. It can also contain more data sets. All of the data sets inside a submission will inherit all of the project metadata that you set up at the very beginning that Luke went through how to set up your project. All of your submissions are within a project and all of your data sets are within the submission. So that's the hierarchical relationship there. Alex said earlier that we do generally suggest smaller submissions. So in case you need to make any changes or if something doesn't pass validation and you really need to push something through, you might want to subset that out from your larger submission. Each submission should generally be of the same level. So maybe you have imaged many specimens in different ways. We generally ask that you subset your raw data or maybe your more processed data into 2 separate submissions. So big takeaways from that are many data sets can make up a single submission, and each data set represents a single donor, individual, or experimental unit. So here's an example of how you might structure your data. At the Brain Image Library, we do have so many different types of techniques and modalities that we accept, and we understand that your experimental design is going to be a little different, and so your images, of course, might not line up to this structure. This is maybe a more common data structure that we see, whereas you have one submission with multiple data sets in them. Those data sets include different image stacks from different channels used in the imaging process. And so, of course, this might not be applicable to your data, generally applicable to what you have. And I do encourage you to reach out to talk about how to structure what you'd like to submit. Best practices are generally that, we do suggest doing a general file cleanup after you've transferred your files. So Luke showed how to do that on demand. Really what I'm looking for there is to make sure that there's no temporary files that kind of got uploaded, or you uploaded what you thought you wanted to, there weren't any extra files included. Also consider that file format is important because we are an open, we do value making sure that the data that you contribute is easily accessible to everyone. So we may ask that you transform any files that are in proprietary formats to an open format, such as what we really recommend is usually a TIFF file. And if that doesn't work for you, please let us know and we can try to figure out what the best format for your images are. That cleanup that you perform after your data transfer might include cleaning up file names. So we do ask that you avoid including spaces or things like that in your file names. Generally, any numbers or letters are fine, including underscores, dashes, or periods. And so the next point here is to create a unique directory for each data set. So really, what's important for for me as a data curator is truly that we are able to connect any piece of metadata that you upload to the exact directory that we're referring to. And so it's very clear that the connections between the metadata and the files that you upload, there is no room for interpretation there, or guessing as to what this metadata is, what images this metadata is trying to describe. And so that is why you can have many data sets within a single submission. And all of these data sets have unique metadata entries. Like, so we do want it to lead to not necessarily not an individual file, but a directory that contains all of the images that are associated with that piece of metadata. And usually those images are going to be only the image files. If you do have additional files that you want to include with your data set, we do ask that you create a new subdirectory that's called extras that includes any other type of information that you want to include with your image data. And are there any questions on any of this, or would you like to see anything that maybe I have spoken about? If not, we do have a place for the break right now. We can take a short 15-minute break and come back at 2 o'clock. And then after that, we'll talk about metadata. I would talk about the next topics first, but the metadata actually can go on for a little bit longer. So I don't want to make anyone wait too long for that. So the plan right now is to meet back at 2 o'clock. All right, thanks everyone. right, everyone, welcome back. My name's Mariah Kenny. I'm the data curator, and I'm going to talk to you all a little bit about the metadata that we collect at the Brain Image Library and how to structure that, how to share that. So if we look at an overview of the submission here, we've discussed how to create an account, which you all have. We've requested access to the portal, created a new submission, and we talked a little bit about uploading the data. But you'll notice that you can simultaneously upload your metadata via the submission portal. And so, you know, it really doesn't matter. It really doesn't matter which one you do first here. It probably takes more time to upload your data to the landing zone than it does to upload your metadata, but you will need to think about these at the same time because you need to think about the structure and record that structure in your metadata file. So that's why these two happen simultaneously. And the real goal of this metadata is to make sure that the microscopy data that you're sharing is able to be reused and is useful to the scientific community, that it contains all the information that someone would need in order to clearly understand what you're sharing. And that really is the goal with sharing all scientific data, right? So we want to make it sure that other people can easily interpret it. And our metadata schema is designed with sharing in mind. It's designed to be able to issue DOIs easily, and we are hoping to make that automatic instead of by request in the future. It is a spreadsheet, so we're all familiar with spreadsheets, so that you can easily change things, or if you need to insert a lot of metadata at one time, you can do that in a spreadsheet, right? And the models that I'm going to go over allow for any redundant fields to be removed. Also gives us a clearer picture of what is linked. And this metadata schema was developed and is described in this preprint. If you want to read more about how this was designed and all of the technical details that go into it. But just an overview of what this metadata that we're asking for is. First, of course, there are contributors who should be given credit for what you're uploading. This can be individuals or it can be groups. So if you would like to cite an entire lab, you can do that using this metadata schema. Funders, any grant that provided financial support. So there is a difference here. The funders that are included here, you can list many. For whenever you were setting up a project and you included a funder ID there, that grant number is usually the one that if you're BICAN related or Brain Initiative related, that project funder ID there should be the one that's we can use for reporting information, like NIH reporting. And these funders, you can include a little more information. You can include any funder that was associated with this project. Then there's publication information, any preprints, protocols, or articles that are associated here. You can also include links to if there's another data set that's published elsewhere or things that are published outside of BIL, such as data sets or even code that you want to, that you maybe published in, Zenodo or something. You can include links to that here. Instrument information defines the instrument that was used to capture the images. Data set information such as titles and abstracts, directories, where to find it, and all of that. You'll include information on the specimen, the donor, organ, sample IDs, and things like that. For 5 of these metadata models, there is an image tab that includes resolution information, orientation information, different information on if there are different channels used, and all of that good information that you need to in order to reuse an image. And then we have also recently included a new metadata field for SWC, or these neuron tracing data sets that are in this standardized SWC format, because the neuron tracings tend to are of this standardized format and are not truly images. We do try to make sure that our metadata is really descriptive of what we have in the brain image library. And so because we do have a growing number of these neuron tracing data sets, we have developed this new metadata schema for it. So whenever you are uploading, you will choose the metadata model that you are uploading. It describes the structure of the submission, really just what's included. Are they all from the same specimen, or do they have different specimens in them? This ensures that your data is properly ingested and indexed. It won't be able to be uploaded. You will see errors if the model and the metadata file don't match. And so let's get a little bit into the details on these 5 models and what they include. So Luke did show you earlier that the landing page that you get to whenever it's time to upload your metadata sheet. We will go and show an example of how to upload this, but whenever you look at this page, there are 5 different options here. You will need to choose one of the 5 metadata models that properly describes your metadata, then choose the associated collection here in order to upload. And so let's take a little more of a deeper dive into what each of these models are. The first is, I would say, is probably the 90% of the brain image library data sets use this metadata model. It is that you have multiple samples and multiple donors, but each data set is coming from a different sample. These are a lot of the whole brain imaging data sets are coming from this, but you use the same instrument with the same parameters to image all of these different samples. So this description says the lines on the specimen tab and match the dataset tab, and each tab should have a corresponding image tab. And there's one line on the instrument tab. So let's look at an example of what that means. So if let me make this full screen and maybe a little larger. So this is the metadata sheet. So the first tab is the README. It includes information on how to fill it out, what's required and what's not, and just some other general information about the specific fields. So what's going to be the same across all of the metadata sheets are the contributors, the funders, and the publications. So you can include here multiple people, of course. For me, I just have myself listed here. Everything starts below. There are 2 examples in these sheets, and those examples should stay there, and you will enter your information underneath all of this. So I've got my name, last name, first name. Am I a creator? So was I involved in producing the data, and should I be listed as an author, really, of this data set? Author in the publishing lingo there. And so, yes, I do want to be listed as a creator, and contributor type, for me, these are all controlled values here. So in the newest version of our metadata sheet, you can choose from the drop-down list here of what your role is. You can also type it in. Then for me, this is a personal contributor type. I'm not an organization. I'm not listing my lab or anything. So it's personal. For those, you need to list an ORCID ID. This is required. Everyone who is listed as a contributor must have an ORCID ID. Then the ORCID, you can have an ORCID ID. There's other types of identifiers, right? ORCID is definitely the most popular. If you're not familiar with ORCID, it is free. You can, it is generally simple procedure to make an ORCID ID. So we do require that everyone has one. Then affiliation, I work at the Pittsburgh Supercomputing Center. So that's my affiliation. This is the ROR for that organization. So the identifier for that affiliation. And then because it's ROR, that's why that's listed there. You can also use GRID or RRIDs for affiliations. But definitely the most common for these two fields are an ORCID or an ROR. So contributors will stay the same for all of the metadata fields or metadata models. Funders is also going to stay the same. So for this, I just have listed the NIH and the ROR link for the NIH as an identifier. And of course, that is an ROR, so I include that as the scheme. Next is the award number and the title for that award. So pretty straightforward there. And then the last one that is the same with it for all of the metadata models is the publication tab. The publication tab includes, you can really cite many different things on this tab. If you have a preprint, you can have the link to like archive or wherever your preprint is. For me, this is a DOI, so I have it listed as a DOI. You can also include any other type of identifier that you have there. You can say it is cited by, because that's what this is. There's other options of what that is for, but is cited by is definitely the most popular one, and then the citation for that. And so the next few tabs are what differs between each metadata field. So if I go back to my model, I know that I have one instrument, many samples that correspond to many data sets that correspond to many images. And so looking here, I can see that I have a single instrument, a confocal microscope. I describe the type and the maker and model. You can include other information here as well that's optional, but I do encourage you to provide as much information as possible about what you have so that it's easily reusable for other people, such as objective, remote magnification, illumination wavelength, things like that. And for me, I have 4 different specimens in here. So I have 4 different data sets. So these are the directories for where each of my data sets lives. This is, it looks like some confocal imaging of these 4 different mice. So those are the titles. It is important that each of these titles is unique. The licensing, most things are in the Random Image Library are CC BY 4.0. The general modality and connectivity, so there are different controlled values here to use. If none of those were truly descriptive of what you have, you also have the choice for other to list the modality that you have. And then an abstract. This abstract should be at least a few sentences. It can be very similar to the abstract that you have if this is related to a preprint or anything like that, but it should be specifically descriptive of the data rather than the experiment or the larger preprint that's associated with these, but it can be similar. So I have many data sets. I have 4 mice that are associated with that. So this is what I mean when I say there's 1 line in the instrument tab and there's 4 lines in the specimen tab, right? And each of these, so line 7 on the data set tab correlates to line 7 on the specimen tab. And so there's specific required information here. We have a local ID, which is really the ID for the donor or the organism, the mouse in my case here. The mouse, the NCBI taxonomy ID is listed here. How old that is, in what units, the sex of the mouse. If you have any genotype information, if you have any other IDs for the organ or for the sample that was taken, this includes those here. It really helps us to be able to link any other data sets that maybe you're going to include some raw data and you're going to include more processed data later. These samples are going to help us link things in the future. And then you can include information on location as well. And then for the image tab, it's the same thing. So everything on line 7 here correlates to all the information on line 7 on this tab. You have orientation information. These are all controlled values again. And a lot more information that a lot of this is optional, but once again, please include as much as you can so that other people can reuse and interpret your data sets. Channel information, display color, dimension information, things like that. This is the newest tab that we have included. So this is the SWC, and because I chose model 1, this tab does need to be here, but I can leave all of this completely blank because that is not the model that I have chosen for this. So that's an in-depth look at model 1. I won't go into detail for all of the other models. For the second one, it's for some data sets, it makes sense to have multiple samples that are all associated with a single data set. So if your experimental design is such that many specimens are imaged, or if there's some type of composite image, or you cannot decipher or tease out specific samples from a particular data set for some reason, you would use model 2. And so there's multiple samples listed in the specimen tab, but there's only one entry for data set, image, and instrument. This is probably one of the least used models because we really do try to ask if you can specify which samples are coming from which images, that those are organized in such a way. The third model is if you have one specimen, but you're looking at unique regions of interest from a specific specimen, but you can do that. There's one specimen, multiple data sets, multiple images in that, but only one instrument metadata field for that. The fourth option is that all of the data sets were generated from a single specimen, but they use different machinery parameters. So, if you were looking at different magnifications, maybe at the same region or same specimen, then you would maybe want to use this one. So you have a single specimen, a single sample, many data sets, many images, and then many instrument parameters listed. The last option is the newest option, and this is for the neuron tracings. So if you are providing neuron tracing in SWC format, you can use this option. It does not include anything on the image tab. So you'll have one entry on the instrument, one entry on the data set, one entry on the specimen, and then many entries on the SWC tab. So let's take a look at that one because it's a little different than all of the others. All right, let me. So this is the same. It has the same contributors, funders, publications. This always remains the same for all of the different models. You would have one entry on the instrument tab, one entry on the data set tab, and one entry on the specimen tab. Your image is completely empty. But then if we look at this, this tab, you'll have many entries. So these really do point to specific files, the specific SWC neuron tracing files. If you have source data, so the images that these tracings were taken from, you can, and that is at the brain image library, you can include that link to the data there. And then if it's from BIL, you can include the sample ID and the submission ID just so that we can easily link all of this information. The next information is SOMA coordinates. And if these coordinates are registered, the region or the ID from the atlas for the name or location of the SOMA. These are not required. These are required if it's registered, but it's not required if it's not. Any ID for the atlas that was used and the name of that atlas. And then some more information. These all have descriptions. If you have any questions about any of these, please let me know. We have information on the segment tags. If you have any there's a standardized format for all of these based off of the community standard that's published. And if you have any deviations from that, those should be included here. And then approved reading level if these are edited or unedited tracings. And then any notes that you would like to include associated with these tracings. Oh, I stopped sharing. Let me, one moment. And so that's what the neuron tracing model looks like. There's more information here on the standard that is accepted within the community and what is what we ask others to use. And so we'll just go through a few quick examples here, just to give you an idea of how of what the questions that you would need to think about in order to say, what does my data look like and how am I going to, which model am I going to use? So if I have 8 MIDGE files from 4 different samples or 4 different mice that are looking at the same part of the brain using the same different microscope setting, I would choose the first option, right? So they're from a single donor and they're on the same instrument. And I can include all of those in the same submission. And so if I was going to look at how to include those and organize those to upload, I would create 4 different directories for the 4 different mice that I have. And those would be my 4 separate data sets. I'll skip the example data sets just for the sake of time here, but for another example is to look is if I have an image taken at 2 different magnifications from the same mouse. For that, I would use the 4th model they're all generated from the same specimen, but use different machinery parameters. And so if I have 3 different images from those 2 different magnifications, maybe these are for an image stack or something like that can be imaged all together, I would include those 2 different magnifications as 2 data sets within my submission. And then for another example is if you have 4 images of 4 different regions of interest from the same brain, you would create 4 different directories for each of those regions of interest. Sorry, that would be metadata model 3. I think I'm a little, I think that was pointing to the wrong one, sorry. And if I have an example where I have multiple samples that are included that have unique metadata, but they can't be separated into specific directories, this is more common, more higher level process information, I would only include those as a single directory here for my data set. Are there any questions about metadata so far? You know, it is really dependent on your technique that you use to generate your data. And so I am always here to answer questions about this. It might be, if you're not at the point where you're ready to submit, you know, this might be a little more, too much to think about right now. But this is, I'm always available to kind of help you figure out how to structure your data sets and structure the metadata files. And these metadata files are curated. And so if you're ever unsure about how do I figure out how to structure my metadata file, I'm always willing to help you fill that out and figure out how to share your information. Any questions, any concerns? With that, I will hand it over to Luke, who's going to show you how do you actually upload this file.
Hey. Sorry, just getting my things together here. Okay, there we go. Okay, so with all that metadata information, it's at this point in your submission process, you would be ready to upload metadata and submit things for validation. So we're going to go through the process of actually uploading a metadata sheet. Mariah dropped a GitHub link in the chat. If you can all go to that and take a look at there's a directory here called metadata model 3 examples. We're going to be working with metadata template v3 model 1. So just the first option here, just click that. And then up top right here, you can download the file. So we're going to go ahead and actually do the metadata upload to that submission we created earlier. Within the BIL submission portal, there is an option for new and then descriptive, or sorry, it should be new add metadata. And I'll show you all where to go ahead and drop this file. So on the portal here, this new and then add metadata. And then we're going to go ahead and we're going to be using metadata option 1 here for this metadata spreadsheet that Mariah provided. And on this drop down, we're going to choose the metadata submission or the data submission that we created earlier. Mine was called Summer Workshop Submission. So for this example, I'm just showing you what happens when you upload a metadata file. To get the metadata template, if you're using Excel, just click this link here. It'll download an empty metadata template to fill out. There's an option here for an ODS file if you're using Linux. So for this example, I just chose option 1, selected my submission here. I'm going to upload metadata and choose that file that I downloaded. I'm going to click open. It's going to prompt and say that it's taking a second to upload all the metadata. This is going to move over to a screen that's going to show all the metadata for all the submissions that you've given to BIL. So mine's going to look like a big jumbled mess because I've uploaded a lot to our website. But you should have a pretty small list here. You can see here under metadata version 2, the submission that I created earlier, Summer Workshop Submission 1. You can go over to the right and click View More to get a more detailed view. Essentially, just kind of takes a spreadsheet that you uploaded and puts it on the screen in a larger format, so you can see all of the contributors, funders, publications, all the different tabs of the spreadsheets that Mariah had gone over. And I'm going to jump back over to my presentation here. So at this point in the process, we've uploaded data, we've uploaded metadata, we've done everything we need to do. So if we're ready to submit our data for validation and publication, this would be the step to do it. To let us know that you're ready to make your data public, there is an option on the portal called Submit Validation Request. This will navigate you to a screen with every different data submission you've ever created on the website. You can check them off, and then there's a button for Submit Validation Request. I'll show that here. So on the BIL Submit site, up on the top right of the bar here, submit publish request. And I'm going to find the submission that I created earlier, mine's Summer Workshop. I'm just going to choose that. You can choose multiple if you're somebody who's doing multiple submissions per quarter, or however you like to do it. It's all about the workflow that best works for you. So you choose this and then submit validation request. What this does is it sends a ticket over to BIL and lets us know that you're ready to make that public. At that point, it goes into the validation process. So what exactly happens during this validation process? So once you send over the request that you're ready to make things public, we run it through a somewhat verbose validation system that does checks on all the files you've uploaded to see if they're empty files. It'll check and fix directory name structures, like if there's, for example, spaces or characters that would trip up a file system, like a comma or some sort of character that would, present as a bash command or something to a file system. It does the same for file names. The images themselves are put through the bioformats program to ensure that the images are usable. And what I mean by that is we're not basing it off of quality of image. It's just a matter of, is the image, is it not just a black screen? Is it not corrupted? Just to make sure that, the images are something somebody can download and actually look at. At that point, the metadata is curated by the data curators on our team and confirmed to be a match with the data submitted. Again, we do not reject based on data quality, only on usability. And as Alex mentioned earlier in the presentation, the process timeline is dependent on the amount of files and the size of the files that are given to us. I just showed you all how to do this. This was submitting that request to us. And at this point, after things are ran through validation and curated, we go ahead and create a public directory for you after all those things are checked off. And again, if you're expecting a very, very large deposit, plan for some data transfer time as well as time for the data validation, and just coordinate with us if you are ever fearful that that's going to be an issue for time frame. We're always happy to work with you. Occasionally too, with the metadata provided, the curation team may reach out and just ask for your assistance with clarification or resolving an issue that doesn't add up with, the structure of data and whatnot. I did want to show lastly here the PI dashboard that's available for project PIs and data managers for projects. Like I mentioned before, when we were creating a submission, there's the option for project. A PI can actually create these projects for you. We can do them internally, but we prefer that a PI is on board and aware of what's going on with the data you're submitting. As the PI, you can create projects, manage projects. If you're a PI or a data manager who's overseeing several different projects and different users uploading, you can get a whole view of everything that's going on underneath you. I'll show over here an option for the PI show over here an option as a PI of what you're able to do. So I have the PI dashboard available for me. If you are a PI or data manager and you wish to have access to this dashboard, just send us a ticket to BIL support, letting us know that either with confirmation from your PI, if you're a data manager, or the PI themselves, saying that here's the projects that I oversee. Am I able to get the access to oversee everything through the BIL submission portal? If you're a PI or a data manager, you can choose manage projects. Here, if you had multiple different projects, as you can see, I only have one here. But if you have multiple different, they would all be listed here. You can view the personnel of the projects. See, I have a few folks internally here that are allowed to make submissions underneath this project name. As a manager or PI, you're allowed to add users to this project. So other people that are submitting to BIL, if you want to give them access to submitting under your grant, you can do it that way. Creating a new project is an option as well. What you would do is provide the name of the grant as well as the grant number for the award, and choosing all that apply for the consortia affiliation. We only have a few options here now. If these options don't apply to you, let us know and we can add those options so that this is accurate. And again, as the data manager or PI, you can view all of the different submissions done underneath your projects. This is just one, and they're just kind of a bunch of test things. Again, if I had multiple, I'd be able to see everything. Same with the management of project users. Same idea here. You can take a look and see who's able to do what under you at BIL. Sorry, there we go. Just want to talk about a couple future features that we plan to add to the submission portal. Some of them not listed here include a downloadable report specific to your grants and projects, as well as we are looking forward in the future for things that program officers would be interested in having for a specific view for themselves. We plan on having the data validation process shown before be something you can prompt yourself as a user and watch as it moves along the way, as well as creation of sub-projects under projects through the PI dashboard, and just for more granularity and specification. When these changes do happen, and they will, we will, of course, keep you all in the loop via the mailing list, and let you know of all the new exciting things that we're adding to these gateways to BIL. With that, I'm finished with my presentation. At this point, you would have a public data set at BIL. And I'm going to hand it back over to Mariah, talking about data citations, DOIs, etc. Unless there's any questions on my end. If not, if Mariah is able to go, she can.
Sure. Let me just share my screen. All right, so let's talk about how to cite your data sets, because that's one of the main goals that you all probably have of depositing your data at BIL, is that you want to cite it, you want to share it, you want to put it in your publications. And so I'm going to talk a little bit about the best practices on how to do that. So we are transitioning to a DOI system for data sets. For data sets that have been issued a DOI, the landing page will contain the citation and the DOI for each data set. You can currently, before we can automatically issue these DOIs, you can reach out to the BIL help desk at any time to request a DOI for the data set. The only thing that we don't recommend you doing is including the download link for your publication. So I'm not sure if this has been shown yet, but anytime you publish a data set, it does get moved to the download .brainimagelibrary.org, and so you can access your data sets here, but this goes directly to the data. And so we will also, people need to know who published this and what's all the metadata included for it. And so we don't suggest including this download link in your publication, or including a query in your publication, such as all of the data sets. Sorry, one second. All of the data sets in BIL for grant X. We want to make sure that what you are citing in your publication is directly accessible, that you have a link for everything that you want to include. And so, we talked quite a bit about what the definition of a data set was, and each data set does get its own DOI. Let's take a look at what that looks like. So every data set has its unique set of metadata. And so that includes the title and the abstract and all of the metadata that goes along with a specific image set, and then the download link for that data set here, including all of the contributors and funding information. And so each data set will get this landing page, but we also can issue a DOI for a group of data sets. So this is probably what you would want to include in your manuscript or article, is a DOI that includes many data sets. And these data sets can be from many submissions. You can have submitted these at many different times, or over the course of a year, if you're continuously uploading data sets to BIL. We can cite all of those data sets at a single time. And it looks very similar, but it does include many links for the data set at the bottom. So this metadata is a little more specific to what is your article about, and maybe why is this group of data significant and how is it related, as opposed to a data set information, which is really just truly what is this data set. So if you would like to request a DOI, we do ask that any group DOIs or any DOIs that are associated with an article are requested via BIL support. You can just send an email to the ticketing system that says, I would like to have a group DOI for my data sets. I will then send you a form that gets the main information that's going to be displayed on the landing page for that grouped UI, information about the publication, and information about all the separate data sets that need to be cited. And while we're in this interim period, before we're automatically issuing DOIs for data sets, you can also reach out to this same BIL support for help getting a DOI for an individual data set. And let's talk a little bit about data access. And once your data is published, where can you find it? Where can you tell others to find it? So there's a few different things. First is the BIL file system, which I showed you a little bit. I'll show you a bit more of it. We have the BIL search portal, and then of course you can point others to DOIs in order to find your data. So whenever you have published your data sets at BIL, the data are organized by submission. So the path to the public data utilizes that submission ID number, the same number that you were using to point your data sets to in the landing zone. That's the same number that gets utilized whenever we publish data sets. It includes there are subdirectories at BIL data, so that correspond to each submission. So if A, B, C, D, E, F is the first few digits of your submission ID, it is AB/CD and then the submission ID, and then any subdirectories for your individual data sets within your submission. And this can be directly reformatted into a URL access. So download.brainimageLibrary.org. If you click on this, this is an example data set, but it does lead you directly to all these images that you can then click on and download if they are a size that you would like to download on your local machine. And then the second way to find public data at BIL is actually using our search portal. So it's submit.brainimageLibrary.org/search. With this, you can see a kind of tracker of what we have available, and then some basic information about data sets. This does include, links to where the data is on the actual, the machine, the file system, and then also the URL link. One of the more popular ways of interacting and finding data sets at BIL currently is actually we have this download inventory option that shows some information. You can download an inventory of what's currently available at BIL. So let's take a look at that. So if I were to let's go to brainimagellibrary.org. And then if we go to data access and then brain inventory, we can take a look at this dashboard. And this download metadata file will actually open up a larger inventory report. It includes metadata version. I'll talk a little bit about that and why some data sets might look a little different. The ID, this is the submission ID, contributors, affiliation, grant number, the link to where this all is located, title, abstract, and other information. So you can get, if you'd prefer to interact with the metadata in a spreadsheet way, then you do have the ability to do that. And let's see. And then, of course, the third way to find data sets at BIL is to know that if you have a DOI for them, this is really the way that we are encouraging other people to share their data, is to really get a DOI for either the group or the data set, so that you can easily see exactly what is is related to each data set. The metadata and the links to the data are all very clear on the DOI landing page. And if you are on the search portal and you're noticing that some of these, or if you're looking at the metadata sheet and you're noticing some of these say version 1 and version 2, and they include different information, we did update the metadata version at the beginning of, not the beginning of this year, it's actually the beginning of last year. And it includes many more fields and a lot of more extra metadata than what was included in version 1. We're in the process of updating all of these to the newest version when possible. And so if you are looking for things and you're noticing that it maybe doesn't have all of the same information, that might be wise, because we do have 2 versions of metadata. And in this period when we're updating things, not all of it might be available. And so are there any questions about finding data or citing data before we move on to this last portion? That I'm just going to talk a little bit about what tools are available, how you can interact with these data sets, because at BIL, some of them are 100 terabytes and you might not want to actually download the data sets and interact with it, or maybe you have large amounts of data that you need to transform before you actually share them. And so at BIL, we actually do have resources in order for you to do exactly that. If you need to do any pre or post submission data manipulation at the Brain Image Library, we do provide resources in order to do that. These are all resources that are available to anyone who chooses to sign up to have an account with the Brain Image Library. You get access to all of these resources for free at no cost to you. The first is the Brain Image Library Analysis Ecosystem. It's made up of several large memory machines, modern GPUs, and is really where you can easily interact with all of the data that you upload to your landing zone, if you need to do any computation associated with that. There are also 2 other machines that are both located at the Pittsburgh Supercomputing Center, which is where the Brain Image Library is based out of. There's Bridges 2 and NeoCortex. Bridges 2 is a supercomputer located at PSC that all the Brain Image Library data is actually mapped to, so you can easily access without needing to download or without needing to transfer any files. You can access all of the Brain Image Library data on Bridges 2. Likewise with NeoCortex. NeoCortex is equipment that is more targeted towards AI development of algorithms or graphic analytics. There are multiple tools that are available to access these resources. First is Open on Demand, which we spoke a little bit about. And we used it in order for you to really take a look at what files you had uploaded, but Open on Demand is actually a much more powerful tool, and that it's a much more powerful tool that gives you the possibility to do a lot of different things. Let's take a look at it real fast. If you want to use Open on Demand, you can do ondemand.psc or ondemand.bil.psc.edu, and it will take you to a page that looks like this. There are a lot of options for you here. So if you want to run any scripts or using Jupyter Lab or RStudio, you can do that on demand. We do have a another workshop that does go more in depth on how to use all of these resources. I'm not going to do that today. I just want to let you know what you have the ability to access. So of course, there's Jupyter Lab or Studio that you can run if you have different scripts. If you have another script that you want to run on terminal, this is basically a terminal for the BIL machines. Let me go back here. And then there's also a way to look at, any file that you have. If I wanted to look at BIL and then the landing zone, we were talking about using this. This is my directory, right? So you can go and look at the different files that you have. If you want to create a new directory or create a subdirectory or a new folder within one of your submissions, you can do that here. You can delete things here if you're really not as comfortable using terminal, but you can also open a terminal here in this directory in order to move things or delete things or anything like that. So truly what I wanted to show you is that you can use RStudio or Jupyter if you wanted to do any pre-processing of your data sets on demand in a way that you don't really have to. This is all based out of your internet browser. You don't have to download any software in order to access this. And then, so that's Open on Demand. Open on Demand is available for the brain image library analysis ecosystem. It's also available on Bridges too. Then there are also other tools such as X2Go or TGX, and these are remote desktop software that give you remote access to the brain image library resources using a graphical user interface. A minute, I have a drink of water. And I'll show you a little bit of X2Go now as well. So X2Go is probably the one that we have documented the most on the website. So if you go to brainimagellibrary.org and you go to data access and then analysis ecosystem, we do have some instructions on how to access the analysis ecosystem. And then it does have specific instructions here of how you can download X2Go and use it. And I'll just show you very quickly what that kind of looks like. So these instructions are provided on the website. And so if you want to connect to X2Go, you would log in. Your login are the same credentials that you use to log into the submit portal, all of that. So I'm going to make this a little bit bigger while it opens up. Give it a moment to load. Oh, that's okay. And so this is actually a virtual machine that's connected to the brain image library analysis ecosystem. So if I click here, I'm going to open a little terminal. And so this is just one way to do this. There are actual many ways to do this. If I wanted to look at, the software that I have to access this, I'll do module avail, and then it'll list some of the software that's available. Of course, if you have software that you would like to use, that you would like to process your submissions with, please reach out and we can help you get access to what you need. If there's or if you have a license to some type of software that you would like to use, we can discuss what the options are there. But if you wanted to do something like Fiji, so I'm going to do module load Fiji and Fiji or image J, whatever you'd like to call it. And so I'm just going to open up Fiji here. And so I think that this is really powerful for you to just do some quick visualization of your data to make sure that what you uploaded is kind of what you're expecting. Or if you need to check something quickly after you've uploaded it, you know, you can go and open up Fiji. I'll give it a second to load. But this is connected to, you know, the brain image library file system. So you can go straight to your landing zone and see exactly what's there and just point and click and open up any files that you have. Still opening. 1 moment. But once again, all of this really is documented on the website. And there's a lot of information here on how to use all of these tools. We generally do a data exploration workshop as well throughout multiple times throughout the year. So if you're interested in how do I use all of these resources and how do I really interact with my data more, though we do provide workshops for that. And if you have any questions, actually, Ivan is here and he is the BIL software specialist on the team, and he will really be able to give you all of the information that you would need. So if I've opened Fiji and I want to, you know, just take a quick look at what I've got going on in the landing zone, I'm going to go, so I'm at BIL landing zone, and then I can go to my directory here, and I don't think I have any files, but I don't want to look at anyone else's stuff, but it's just as easy as going, clicking, and pointing at what file you want to open and just visualizing like that. So just to give you an idea of what is available. So that is X2Go. GTX or TGX is a remote desktop similar to X2Go that is a little more graphically intensive and can get higher resolution. But just to give you an idea that of the analysis ecosystem, it is an HPC high processing HPC system. There are, you know, many terabytes of RAM, many nodes, and if there's any type of pre-processing that you need to do, there is a lot of processing power here in order to do that. There's many different software available. This is just naming a few Fiji, bioformats, R, Python, MATLAB, whatever you might need, we can try to work with you to see what you need. And then Bridges 2, just to give you a small idea of what Bridges 2 is, if you need to do anything that is particularly, you know, very computationally intensive, or is maybe going to take a longer amount of time, or maybe it would use all of the brain image library resources, there is Bridges 2 as well. It is available at no cost to researchers, and there's more information here at psu.edu/resources/Bridges 2 to figure out how to get access to Bridges 2. All of the files on the BIL file system are mounted on Bridges 2, so it's directly accessible. You won't need to download anything or change do any type of file transfer in order to interact with your data in a larger or more computationally intensive environment. And if you have any questions on anything really that we've mentioned today, one, feel free to ask them now, but also we do have office hours. They currently occur on the second Wednesday of every month from 2 to 3. I'm present at these office hours. Luke is usually there as well. We can pull in other people as we need if you have specific questions, but they do occur monthly. If you click on this link, you can see the exact day and time, and whenever you register for this, it'll send you the Zoom link, and it will also provide you with an opportunity to add it to your calendar. If you would like to register for this, but you don't want to go to every occurrence, that is totally fine. Just to get it on your calendar to see if you can go, if you need that at the time that you're working on all of this. So if you can't make it to those though, you can always contact us at BIL support. If you would like to set up, we're always available to set up different a virtual, meeting to kind of walk you through everything, or if you're experiencing any difficulties, we're here for you. Are there any questions on anything about the resources or the submission process that anyone has any questions on? That is the end of our program today. I mean, if anyone has any, I'm going to stop the recording. And